Okay, I would then introduce FrancescaCardinale, who is Associate Professor at the University of Turin's Department for Agriculture, Forest and Food Science, the same that Andrea works in. Francesca's research focuses on molecular plant physiology, on biotic and abiotic stress perception, on signal transduction. And she works on phytohormones strigolactones, and Francesca leads the work package 2, the second one, TOMRES, that Andrea introduced right now.
Within work package 2 of TOMRES, we've been dealing with a number of different aspects related to strigolactones and their role in water and nutrient use efficiency under single and combined stress. Today, I chose to talk to you about unpublished data, so it's a bit of preliminary, but I thought it would be more interesting for the TOMRES people than hearing what they. Are you changing my slides? Oh, because
no, I can't change the slides
by itself. Good. Well, anyway, so what are strigolactones? They are a family of molecules that are that share a tricyclic lactone structure connected to a fourth eno-ether bridge by to a fourth cycle by an eno-ether bridge, and they're produced from carotenoids in the plastids, where they are first isomerized and then further cleaved and rearranged by 2 carotenoid cleavage dioxygenases named CCD7 and 8. This leads to an intermediate, which is carlactone, that is then released from the plastid and modified by a diversified enzymatic pathway that is slightly different in different plants, so that a blend of molecules is produced that is specific for that plant. From the roots, where they are mainly but not exclusively produced, strigolactones are then either transported shootward, where they act as hormones, as we will see, or they act locally, or they are exuded in the rhizosphere, where they interact with the existing microflora and other organisms. So for about 40 years, they've been known only as components of root exudates, but then in 2008, they have been associated to a hormonal function for the producing plant, and specifically they've been recognized as inhibitors of tillering and branching. A number of other functions in development have been assigned to them over the years, as it's normal for hormones. Among them, for certain plant species, is reproduction, and we will hear about this again later. They've also been associated to responses to environmental stresses, initially especially low phosphate and nitrogen in soil, and more recently also at the shoot level, they've been proven important for a correct acclimation and response to drought. Being at the crossroads between development and environmental stress responses, they are increasingly seen as resource allocators under environmental stress. And this is what is interesting for us today. So I decided, because of the time constraints, that I will start off by presenting you the working model for strigolactone contribution to drought responses. This model has been, has received contributions also from other model systems, such as Arabidopsis and Lotus, but is now based mainly on what we know in tomato. So this is what we think happens around strigolactones and abscisic acid during either no stress on the left part of the screen or osmotic and drought stress on the right part. So we and others have demonstrated that when strigolactones are compromised in signaling or synthesis, the plant is oversensitive to drought. And consistent with a positive role in the acclimation to drought, it is proven that during an osmotic stress event, transcription of the genes in the leaves followed by synthesis of strigolactones will go up. And if we treat with exogenous strigolactones, then we will see that sensitivity to ABA will go up as well. So this seems to be, if not the main, one of the most important ways by which strigolactones positively regulate drought responses. So what happens in the roots in the meanwhile? Underground, we've seen this, that the roots are the main site of production. From underground, the strigolactones will be translocated shootward, and there they will act in a negative feedback loop as inhibitors of local synthesis of strigolactones by acting on gene transcription. So under normal conditions, strigolactones in the leaves are kept low, and sensitivity is also to ABA is kept low, because of strigolactones flowing from the roots and directly or indirectly inhibiting synthesis. What happens under drought, however, in the roots is a bit counterintuitive, if you wish. That is, the synthesis drops. And this is something that is not just metabolic suffering. Why is it going back and forth? It's just not metabolic suffering, because it is needed, this drop, to let local ABA levels increase. This was proven in lotus. We also know from Arabidopsis that this changes the way ABA is mobilized in the tissue and it is transported. Systemically, the less abundant flow of strigolactones can directly or indirectly relieve the biosynthetic pathway in the leaves from the inhibition that happens under regular conditions. So that sensitivity to ABA can go up together with strigolactone synthesis. We know that there's, we're starting to know now a little bit about the mediators of this effect of strigolactones on ABA sensitivity. One such mediator may be MIR156. This is a microRNA that has known developmental roles, and we will hear a bit about it later because of these developmental roles. But here in the context of drought, they seem to mediate the effect of strigolactones on ABA sensitivity at the guard cell level. So this module, strigolactones, MIR156 and ABA, seems to be important for the environmental memory at the guard cell level. This was also part of TOMRES research, but I will not present new data on this today. Rather, I would spend a minute presenting you a grafting combination that we've been using to play with the strigolactone levels in TOMRES as well. And this is a combination that mimics genetically what happens during drought, physiologically, to the strigolactone levels. That is, we put together a wild-type scion to a strigolactone-depleted rootstock. And in this combination, we, in fact, reproduce what happens during drought. And in fact, also the phenotype, the molecular and physiological phenotype of these grafting combination is consistent with the model that I explained before, because strigolactone synthesis is activated in these wild-type shoots more than when the wild-type shoot is onto wild-type roots. And stomata are closer because sensitivity to ABA is higher in these in the guard cells of these leaves. So keep this information in mind, because this is a kind of plant that we've been using also for other purposes, and they can be seen as, so to say, complementary and opposite to strigolactone depleted plants for certain aspects. So we had the task in TOMRES to see whether strigolactones could influence water use efficiency, as it was reasonable to suspect. And we started off by treating with the synthetic strigolactone analog GR24. Initially, we were working with young plants, either seedlings in vitro or potted but young plants in the greenhouse. And we consistently observed that when we were giving excess strigolactone, we would see an increased root biomass, and therefore, when measurable, an increased water use efficiency at the root level. What happens when we look at the grafted plants? Here you see an experiment in which you have self-grafted wild type and strigolactone depleted plants. This is white and gray, and then heterografted plants in black. And the plants are potted in greenhouse and either kept at the 100% field capacity or kept at 60% for 4 weeks, right before the beginning of flowering. And then as the first flower appeared on the first plant, they were let recover and freely reproduce. And what we observed was something consistent with the idea that resources in excess strigolactones would go to the roots, because we had this significant difference that is highlighted in the graph. So where does this lead us? We were also interested in checking what happens to fruits, because this is a relevant trait for tomato as a crop. So we checked for the agronomic water use efficiency, so yield over water, in these kind of plants. And what we saw is that there was a trend, though not significant, for higher agronomic water use efficiency in the heterografted plants. And there was a very significant and dramatic decrease in water use efficiency in the strigolactone deficient plants. Since these plants do not have a general water use efficiency problem, they have a very high efficiency for shoots, for example, for the vegetative part of the shoot. We wondered whether this effect that is highly that is very much worsened by drought could be linked to a specific defect for reproduction. And in fact, there were hints in the literature that this could be the case, because especially among Solanaceous plants, when strigolactones are not working or are not received, they are not synthesized, then we do have a phenotype in reproduction that can be also quite severe, depending on how strong the molecular phenotype is. And also, outside Solanaceous plants, Lotus japonicus is compromised when strigolactone in reproduction, when strigolactones are not synthesized properly. This aspect has been overlooked so far, possibly because the main model species for strigolactones, that are Arabidopsis P and rice, have no apparent effect, so they were kind of neglected. How about tomato? There's a mild phenotype published for a similar line to the one that we use, that is silenced on the next enzyme in the pathway, CCD8, for having smaller and fewer flowers and fruits. Our line had no reported phenotype, but we knew from handling them that whenever they were, we wanted to reproduce them and we were not controlling properly all conditions, especially water, then they would have big trouble. So we decided that we should investigate a bit more what the effects specifically of strigolactones are on reproduction in tomato, because this is so relevant for agronomic water use efficiency and yield in general. We started by treating potted plants and checking their performances during flowering and fruiting. And the answer is, if we treat with the synthetic strigolactone analog, GR24, we reduce the time that is needed for flowering. And we increase yield in terms of fruit number per plant, especially at the early harvesting. And the cumulative yield maintains this difference, this gain of the treated versus untreated plants. So does this treatment also induce any marker of floral transition? We started investigating from obvious candidates first. And the answer is yes. The first and most meaningful, if you wish, to assess is SFT. This is the floral effector. It's the homologue of FT in Arabidopsis, and it produces the florigen. And it's described in the leaves. Then the product travels to the shoot apex to initiate floral transition and orchestrate with other players the production of the inflorescence. Also, we observed that there's a transient and so short-lived induction of another player, MIR172. This is a microRNA. We will see these players in a wider context at the end of this part. And how about grafted plants? Do they have a consistent phenotype, and what happens about molecular markers? If we look at the flowering of the 3 grafted lines that we have seen in the previous experiment, here stress is not included, it's just regular development. We see that there's a connection, there's a correlation between the level of strigolactones. This is the line up here for the heterografted plants with higher strigolactone content in the leaves, and the black bar, which is for the yield in terms of fruit number per plant. The lowest producing and flowering is the strigolactone-depleted, and then the wild type is an intermediate value. So yes, there's a correlation between strigolactone levels and flowering and fruiting. Are the markers that are moving in these plants the same or similar as the ones that we see moving after treatment with the synthetic strigolactone? The answer is yes. We performed a transcriptome analysis, and I will spare you the list of the genes and all the general graphs, but the gene ontology term reproduction is one of the many that are enriched in the strigolactone-depleted plants. And when we go for targeted quantification of specific markers, we see that the strigolactones seem to affect a module that is called the MIR319 lanceolate SFT. So we have a positive correlation between strigolactone levels and 2 promoters of flowering, so the effector SFT and MIR319. Here, unluckily, I miss the heterografted plants, but the trend is confirmed in mutants compared to the wild type. And then there's the opposite relationship, so a negative correlation with the repressor lanceolate. So where does this place strigolactones in the network of the known floral promoting and inhibiting pathways? Here we are starting to clearing our minds about what could be. It's quite preliminary at the moment, but so what we know is that there's a direct correlation with the transcription of SFT in the leaves, starting from strigolactone levels in the leaves. We see that there's an induction by, so that strigolactone levels correlate with the level of a microRNA, which is number 172. And this microRNA is supposed to go up over time while the plant ages and reaches reproductive competence. So this initially was taken as a symptom that the strigolactones could promote and accelerate the age-related pathway. However, MIR172 is supposed to act on SFT via the inhibition of another microRNA, which is number 156. This one is supposed to drop with age, so to relieve inhibition onto SFT. However, as we know, also MIR156 positively correlates with strigolactones. We know this from the drought studies, but we have seen this also in our grafted plants. So this doesn't seem to be the branch in the network that is specifically stimulated by the strigolactones. However, the
After 2 minutes, Francesca.
Okay, yes, I'm done. However, the module that goes through MIR319, lanceolate, and SFT seems to be stimulated. There is also another branch of the pathway that is the one that connects this module to gibberellins. So the phenotype is consistent with a high level of gibberellins in the mutants. Gibberellins in tomato inhibit flowering, contrarily to Arabidopsis. we checked for the signature in the transcriptome, and this is consistent. When we checked for the metabolites, however, we saw a significant trend for more bioactive gibberellins. Not significant, we saw a clear trend for more gibberellins, but no significance for the moment. So we have to be careful in the conclusions here. That will sound like this. We've seen that strigolactones in the shoot promote drought acclimation partly via ABA and a microRNA called 156, and that strigolactone levels correlate with root and agronomic water use efficiency in tomato, and that the strigolactone-dependent resource allocation to fruits seems to be linked to their effects on the promotion of floral transition. And this promotion might go through the module that is articulated around MIR 319, lanceolate, and possibly via a mitigation of gibberellin action. And the final thought is that we are starting to see strigolactones as important, not so much as an emergency measure when drought occurs, but rather to reset the plant physiology to cope for the next steps, either another drought stress or drought escape, for example, in flowering. And this will be it. I want to finish by just posting the picture of the enlarged group. This is pre-COVID, but many people are still in our group. And the ones whose names are in bold are the ones who contributed most of the data that I presented today. And thank you very much.
Thank you very much, Francesca. You need to imagine the clapping of the audience now. Yes. Thank you very much for your presentation.